Hello everyone, my name is Muge Karaman. I am a research assistant professor at the BME department and I've been conducting my research at the Center for Magnetic Resonance Research, CMRR of UIC. CMRR has two locations, one at the Advanced Imaging Center, which houses a 3T fully research dedicated MRI scanner where we primarily conduct our research. MRI is a technique that uses a powerful magnet and radio waves to create detailed images of the internal body structures. The first step in this pipeline is MRI data acquisition, which produces a set of MR images, either anatomical images that the radiologist can interpret directly, or other types of images based on more advanced MRI techniques that have functional information, such as diffusion MRI. Unlike the anatomical ones, these types of images need to go through an additional step for MRI data analysis to produce MR-based imaging parameters. These images are then evaluated by a team of clinicians who make important clinical decisions. At the CMRR, this research for MRI data acquisition is primarily led by Dr. Joe Zhao, who is also affiliated with the Department of BME. My contributions are mostly on the MRI data analysis part. These two research areas, however, are intertwined, and we are collaborating with a team of scientists and clinicians at UIC and other institutions. Today, I'm going to show a few examples of our studies in these two areas. An important challenge in MRI is low temporal resolution, which prevents MRI to capture rapid dynamic events. Our laboratory is active in developing advanced techniques to improve MR image quality. In our most recent work, we were able to produce MR images with sub-millisecond temporal resolution, as demonstrated here on cardiac MRI. Another area that our center specializes in is MRI data analysis, primarily for diffusion-weighted MRI. Diffusion-weighted imaging is used by the clinicians to non-invasively characterize the complex biological tissue, such as the cancer tissue, as it provides an opportunity to reveal underlying microstructural information by measuring the random motion of water molecules. In a typical diffusion MRI experiment, the collected MRI data for each voxel in the image is a signal intensity curve that decays with increasing B value. What's challenging here is to model the signal decay curve to produce interpretable parameter maps. One area of our research is to develop mathematical techniques to analyze the diffusion MRI data to produce maps that reveal additional tissue property-related information, such as tissue heterogeneity, compared to the conventional techniques. We investigated the feasibility of these techniques in a variety of clinical problems, such as pediatric brain tumor grading, where we see significant differences in model parameters between the low and high-grade tumors, also in grading adult gliomas or classifying gastric cancer, or in other diseases. Most recently, we have directed our efforts into developing advanced diffusion-weighted imaging techniques for tissue characterization, breast cancer, and also predicting tumor response to chemotherapy, which is a study that is currently funded by NIH. Thank you for listening. Please email me if you have any questions.